Hello students, this is the third lecture in the series on muscles, and this one is going to talk about the contraction coupling. Sometimes it's called the sliding filament or cross-bridge cycling. Now if you just watch the excitation at the neuromuscular junction, basically we're picking up where we left off. And where we left off, we have the action potential that is now acting on the sarcolemma of the muscle fiber. So the process of this contraction coupling, the muscle contraction, we're going to start with number one. That's where we're going to start, with the action potential propagating along the sarcolemma. Now, what I want you to visualize is that it's spreading from that neuromuscular junction all around the muscle fiber and down the T-tubules. Now remember, those T-tubules were invaginations of the sarcolemma. It was the way the sarcolemma is kind of embedded into the rest of the cell. So instead of just encircling that cell, the sarcolemma has these T-tubules that go deep into the muscle fiber. So number one, action potential propagated along the sarcolemma, flow down the T-tubules. So let's remember that triad relationship. Let's look at this picture here. All right, so if we look at this, the red arrow. The red arrow is representing the dipolar. Sorry, I don't know why it keeps doing that. I can't leave it on there very long. It's representing the depolarization event, that action potential being propagated along that plasma membrane, the sarcolemma. And as it does that, it goes down the T-tubules. Makes sense. Now the triad relationship. I said the triad was 1 part T-tubule and 2 parts of the endoplasmic reticulum, what we call the sarcoplasmic reticulum. They're kind of big swollen areas of that SR. And here they are represented in the picture. Now we have a name for those big swollen areas on either side of that T-tubule, and we call that the terminal cisternae. Cistern, something that holds. And what it holds is calcium. So remember, the major function of the sarcoplasmic reticulum was to store and release calcium ions. So these little red balls that you're seeing represent calcium ions. So as that depolarization event flows down the T-tubules, what it does is that it stimulates that sarcoplasmic reticulum to release calcium ions from the terminal cisternae. Okay? So you can imagine that depolarization event going down, where it's shown yellow on there, going down, stimulating, opening up ion channels, and allowing for the calcium ions to rush out into the muscle fiber. Okay, we're going to basically follow those calcium ions. Now, the calcium ions rush out into the muscle fiber, and they are right there where the sarcomeres are. Remember the sarcomeres? And the sarcomere is that functional unit of the muscle fiber. And within that sarcomere, we talked about there are 2 major types of proteins. There are the actin and the myosin. Both of them come in terms of filaments. So we have the actin filament and the myosin filament. Okay, we're going to track that calcium ion, I said. So we're on to number 3. What happens next? Calcium ions bind to the troponin. Okay, so I'm going to take you right up here on the picture. Here's the calcium ion, and it's shown as a blue ball. The calcium ion will bind to the troponin. Remember, troponin was one of those regulatory proteins associated with the actin filament. The troponin is attached to the tropomyosin. And something else you learned about the tropomyosin is that in a relaxed muscle, that tropomyosin is covering up the active sites on the actin. Okay? So right before that calcium gets released, this is what it looks like. Now the calcium gets released. Here comes the calcium. It binds to the troponin. And sometimes when molecules bind to each other, it causes a shape change. And that's what happens here. So the calcium binds to the troponin. It causes a shape change. In technical terms, we call that a conformational change. But it's just a shape change. And when that happens, the troponin will stimulate the tropomyosin to move away. Okay? So it moves away from the active sites on the actin, and you can see that here. You can see those little black dots here, and that represents the active sites being opened up. Okay, so that's number 3 and 4. Now we have to look at what's going on with the myosin. What happens is that once these are uncovered, the myosin are ready to bind to the actin. So one molecule of ATP energizes the myosin heads. And what will happen is it will pop up and bind to the actin. So that's number 5. One molecule of ATP energizes myosin head to bind to an actin molecule. Number 6. The myosin molecule acts as a ratchet to move the actin filament. So, right over here, it will actually bend in order to ratchet and push the actin molecule, the actin filament. So number 6, the myosin molecule acts as a ratchet to move the actin filament. Here we go. It's ratcheting. It's moving the entire filament. This stays put. Okay? The heads pop up and the heads move them. Now, one molecule of ATP is used to unbind the myosin. That's shown here. We're going to need an ATP molecule to come in and help it unbind. Okay, it's not going to unbind without the ATP. Now, so what has happened essentially is that the myosin filament, the heads of the myosin filament pop up and they move the actin filament. They physically move the actin filament. What that causes is sliding. It causes sliding of the filaments, of the thin filament. So you're going to see more overlap of the actin filaments over the myosin filaments. When you have that overlap, that is your muscle contraction. Now once those heads use ATP to pop off and relax back down, slides back. That's a relaxed muscle. Now after that contraction, we're going to need one molecule of ATP to move all of those calcium ions back into the sarcoplasmic reticulum. You actually need one molecule of ATP to move 2 calcium ions back into the sarcoplasmic reticulum. So in essence, and this is happening on a grand scale, not just one head is going to pop up, but you're going to see many, many heads popping up and helping to move that slide, that filament. On a grand scale, we use a lot of ATP. Just looking at this one model example, we're using one ATP molecule to energize the myosin head. We're using one ATP molecule to unbind the myosin head. And then we're using another molecule of ATP to move only two of those calcium ions back into the sarcoplasmic reticulum. So lots of in a typical muscle contraction, we're going to need a lot of ATP because this is on a grand scale. Alright, I have a very short video for you to watch. A little animation, because sometimes when I'm talking about it and you're looking at two-dimensional pictures, it's very hard to visualize. So the more animations that you can watch about this, the better I think it will solidify in your head. I found a short one.
Sliding filament theory is the mechanism by which muscles are thought to contract at a cellular level. An understanding of the structure of skeletal muscle is useful when learning how sliding filament theory works. Each muscle is made up of a number of bundles of muscle fibers. Each bundle of muscle fibers contains anywhere from 10 and 100 individual fibers. Each muscle fiber itself contains cylindrical organelles known as myofibrils, which themselves are bundles of proteins called actin and myosin. Surrounding the myofibril, there is a network of tubules and channels called the sarcoplasmic reticulum, where calcium is stored. Each myofibril can be broken down into functional repeating segments called sarcomeres. If we look at a two-dimensional model of a sarcomere, it consists of actin and myosin. When a nerve impulse arrives at the muscle, it causes a release of a chemical called acetylcholine. The presence of acetylcholine causes depolarization, enabling calcium to be released from the sarcoplasmic reticulum. The calcium binds to troponin, changing its shape, and so moving tropomyosin from the active site of the actin. The myosin filaments can now attach to the actin, forming a cross bridge. The breakdown of ATP releases energy, which enables the myosin to pull the actin filaments inwards, contracting the muscle. This occurs along the entire length of every myofibril in the muscle cell. When an ATP molecule binds to the myosin head, the myosin detaches from the actin and the cross bridge is broken. When the ATP is then broken down, the myosin head can again attach to an actin binding site further along the actin filament and repeat the process. This repeated pulling of the actin over the myosin is often known as the ratchet mechanism. This process of muscular contraction can last for as long as there are adequate ATP and Ca+ stores. Once the nerve impulse stops, the Ca+ is pumped back to the sarcoplasmic reticulum and the actin returns to its resting position, causing the muscle to lengthen and relax.
Okay. I'm going to back this up a little bit.
Once the nerve impulse stops, the Ca+ is pumped back to the sarcoplasmic reticulum and the actin returns to its resting position, causing the muscle to...
Alright, so I just want to point something out. Alright, so you notice what we're actually seeing right here is you're seeing basically lines, right? These lines are representing the filaments. And I said that the myosin is a thick filament, and that is the darker one. I mean the lighter one. That would represent the myosin filament. And you notice when it contracted, we had more of an overlap of the purple ones, which are the actin filaments. So in a muscle contraction, you're going to have more of an overlap of it. Okay, so if you take your fingers, take your hands, put your pins down, and take your hands with your fingers and kind of interlace your fingers, right? And when they're completely interlaced, that means that there's more sliding that's going on. In a relaxed muscle, there's barely any overlap. So when you watch this...
Lengthen and relax.
She went too far. Hold on. There.
This process of muscular contraction can last for as long...
Okay, so we saw more and more overlap during that contraction. The more overlap, what's going to happen is overall in that muscle, the belly or the gaster of that muscle is going to become thicker. Okay, so if you take your hand and put it on your bicep, okay, you guys know where that is. Put it on your bicep, relax it, and then contract it. Contract it as hard as you can. Right? You feel that belly or the gaster of the muscle getting thicker. Right? Impulses are being sent. spreading along the sarcolemma. It's getting these, you're getting more overlapping, you're getting more sliding of the actin filaments. Okay, and that means here, more overlap means it's going to get thicker on a grand scale. So make that muscle and feel it. I want you to feel it getting thicker. Feel it getting bigger within that belly or gaster of the muscle. All right, now hold that contraction. Hold it. And then try to make it even more forceful. So even more, trying to make it even bigger. Did you feel it getting just slightly bigger? That means you're getting more overlap. The more overlap, the thicker, the bigger the muscle. Okay, and that's part of that contraction. I'm going to go back to where we were. Okay, so this is where we were. That short video was the first link. Now I did find another video that is a bit longer, and I think it would be good. It just gives you a more three-dimensional visualization of what we're talking about, that sliding filament theory of that muscle contraction. This one I know you can't click on because I'm turning this into a video, but I will put this link underneath the video that's playing for the YouTube. So if you just look underneath in that comments for the YouTube, I'll put this video, that link there. Now, just like we talked about Botox and how it related to the neuromuscular junction, there is a phenomenon known as rigor mortis that occurs after death. It is one of the, there are different types of rigor that occur after death in the decomposition process, and this is just one of them. So it's called rigor mortis. I don't know if you've ever heard of it before, but for a certain period of time after death, bodies can take on a very rigid, I shouldn't say just appearance, but the body becomes rigid. And why are we talking about this now? It's because this is where it applies. So what you just learned, actually, we're going to, of course, in muscle contraction, but we're going to apply it to this rigor mortis because now you can understand it very well. So in rigor mortis, after death, a certain period after death, all of the muscles, your body becomes very rigid. And what happens is all of the muscles become contracted. All right? Now you know how that happens now. And take a look at this picture over here. So you know what has to happen. Neuromuscular junction, right? Excitation. You know the whole gamut. You know that the myosin heads have to pop up and they have to bind to the active site on the actin. And then the ratcheting motion, you get your contraction. Okay. You know all that. Now, what else you learned is that you have to have ATP. Okay, there's 3 times, specifically in a muscle contraction, where ATP was used. Can you name them? I'll give you a second. So the first one that I told you about was to energize the myosin head so that it can pop up and bind. Okay? The second one was that we needed the ATP in order to unbind the heads. And then we need ATP in order to shuttle the calcium ions back into the sarcoplasmic reticulum. So from what you know now, if there's calcium out there, you're going to have a muscle contraction. Alright, so after death, once we die, what's going to happen is that the calcium that is in that terminal cisterna of the sarcoplasmic reticulum starts to break down. Okay, so the walls of that organelle starts to break down and releases the calcium into the muscle fiber. Calcium does what calcium does, and it's going to bind to the troponin. Binds to the troponin, causes a conformational change, shape change, opens up the active sites on the actin, and guess what happens to the myosin heads? They've already been energized by the ATP. They're going to pop up and bind. They're going to cause a contraction. They're going to cause that sliding filament, and which is a contraction. Remember in our process what happened next? Yep, we had to have that ATP come in, bind to the myosin head in order for it to detach. Well, guess what we're not making any more of if we're dead? ATP. We're not making any more ATP. The amount of ATP that was still left in the muscle fiber right after death was all used to energize the heads on the myosin. And it did what it did. Once the active sites on the actin were opened up, it popped up, it bind, it slide, got that muscle contraction. But since we're not making any more ATP, they can't pop off. We can't get them off. So now we have this sustained muscle contraction. And that muscle contraction is going to, all of those muscles are going to stay contracted until those proteins, unfortunately, start to break down after death. So once the proteins start to break down, then the muscles will start to relax and the rigor mortis will go away. Alright, so now you know. Alright, so this concludes this particular lecture of muscles. And you're going to have one more short muscle lecture that goes in the series. Alright, so thanks for listening to this one.